session, I'm going to talk about the clinical enzyme usage in medicine. At the end of this session, you will learn about the prescribing medical enzyme to recognize or distinguish some disease. So I want to start with one question: how can we recognize a problem in the body? By the enzymatic activity of some enzyme in blood circulation. So enzymatic diagnosis is a method to diagnose disease by measure the content and changes of the certain substance in the body, or through the change of the original enzyme activity in the body. Enzyme will give us about the disease state due to their corresponding altered pattern in tissue and cell. All enzymes that we have in blood circulation, they are classified in two big groups. Measurement of the activity of enzyme in plasma has value in the diagnosis and management of the wide variety of disease. So about the plasma enzyme, we can have two big classification based on their function in blood circulation. First group is actually functional enzyme. It means they have function in blood circulation, they do something, they catalyze some reaction. Big example and most common example for this group of functional enzyme are coagulating enzyme that they are involved in coagulation cascade. The second big classification for enzyme inside the blood circulation are non-functional enzymes. It means they are not working in blood circulation. They are in our tissue cells, but after activity inside the cell, some part of them will be leaked in blood circulation. The level of the non-functional enzyme can be controlled by actually two mechanisms. One of them it is because of damage to the tissue of origin or proliferation of the cell from which the enzyme arise. So plasma enzyme can be classified to functional and non-functional enzyme. Here you see some differences between the functional and non-functional enzyme. For example, about the synthesis place of the functional enzyme, they are synthesized by the liver, but they are the non-functional. They are belongs to intracellular different body organs like skeletal muscle, heart, brain, and other tissues. About their function, as it comes from the name, functional enzyme for specific function in blood, but about the non-functional enzyme, they do not have physical function in blood. And about the other differences between them, you can see here in one table. Here we have two terms about the enzymes. One of them is proenzyme and the next one is isoenzyme. Proenzyme, or another name that is a general name for this kind of enzyme, is a zymogen. They are inactive form of the enzyme that becomes active after partial proteolysis. Example for this kind of zymogens are coagulating enzyme, or here we have another example that is a digestive enzyme pepsinogen. Converting pepsinogen to pepsin happens by cleavage that we have inside the protein. Here we removed one part of this whole protein, and after that the pepsinogen is going to be activated and producing the pepsin. Pepsin is active form of the pepsinogen. The next term is the isoenzyme. Actually, they are different molecular form of the same enzyme synthesized by different tissue. They act on same substrate. Here, I want to compare isoenzyme similarity and differences between each other. About their similarity, they catalyze the same reaction, act on the same substrate, and use the same coenzyme. But about their differences, their subunits are totally different. So because of these differences between their subunits, so they have different physical properties and result in their Vmax and Km above the velocity and the affinity of this enzyme, this kind of isoenzyme. We will have some actually differences. Their electrophoretic mobility and immunogenetic properties of them are totally different. And final thing that is different between this isoenzyme is a heat stability. So what's the importance of this isoenzyme? Actually, study of the isoenzyme increase the specificity of non-functional plasma enzyme for various tissue. Knowing the intracellular localization of enzyme help us to predict the actually severity of problem or disorder and disease. For example, when we are talking about the lactate dehydrogenase, it means we know the lactate dehydrogenase is distributed inside the cytoplasm. So above the other parts of the cells, like mitochondria, we have more than 70% of the AST inside the mitochondria. But about the rest part of the AST, actually there's 30% of the AST is distributed inside the cytoplasm. So importance of the knowing this intracellular localization of this enzyme is about the severity of the problem. When we have a condition that problem causes just mild problem to the cell, we will have cytoplasm enzyme that they are going to be released to blood circulation. But about the condition that we have a severe condition, severe damage to the cells, it means our mitochondria is going to be actually disrupted and release the enzyme to the blood circulation. But about the other parts of the intracellular location, we have a lysosome specific enzyme. When we have an increased level of the ASP, it means the lysosome of the cells is disrupted. And about the other parts, for example, for the GGT and ALP, they are localized in membrane of the cell. So with mild problem to the cell membrane, we will have increased level of the GGT and ALT in blood circulation. Here you can see diagnostic serum enzyme in one table. Most of these enzymes nowadays are most common form of enzyme in clinics used for the diagnosis specific disease. Here you can see the whole name and the abbreviation of this enzyme. Maybe you see in request for the laboratory by physicians. Here, for example, we have acid phosphatase, is one of the enzyme that is used in clinic. It's abbreviated as a ACP or AAP, alkaline phosphatase as a ALP, alkaline transaminase as a ALT, and the rest of the enzyme you see here with their abbreviation. We will learn about the most of this enzyme because they are important in clinic, and you will face with most of them. So this session we will talk in two parts about the diagnostic and prognostic functions of this non-functional plasma enzyme, and the next application of this enzyme is a therapeutic agent for some diseases. So I want to start with liver function test. You know, sometimes some physicians they want to analyze the liver, so they request for the liver function test. In which condition we need to actually analyze or assess the liver. So liver function test can be used to screen for liver infections such as hepatitis, monitor the progression of the disease such as viral or alcoholic hepatitis, and determine how blood treatment is working measure the severity of disease, particularly scarring of the liver in cirrhosis condition. And finally, we can use liver function test to monitor possible side effects of medications. For liver function test, it is customary and useful to categorize liver diseases into three broad categories. Hepatocellular, in which primary injury is into the hepatocytes. Cholestatic, in which primary injury is to the bile duct. And infiltrative, in which the liver is invaded or replaced by non-hepatic substance such as neoplasm or amyloid in metastatic condition of other cancers to the liver. So for the liver function test, we can have some tests. Some of them is about the synthesis of the some protein by liver, like serum albumin. We can analyze serum albumin. And we have another group of enzymes that they are useful for liver function test. They are called blood liver enzyme. Here we have some of them: alanine aminotransferase, aspartate aminotransferase, and gamma glutamyl transferase, and finally alkaline phosphatase. Most common enzymes that they are useful for liver function test. The next test you can use for the liver function test is the blood bilirubin test and blood coagulation factor. Actually, all of them, they are used in liver function test. But for this session, we are not going to learn about all of these tests. We are going to just to learn about the blood liver enzyme as a applicable actually enzymes to assess the liver function. So in this session we will learn about the blood liver enzyme for the liver function test, but about the other tests you will learn in other sessions and topics. So we have two enzymes, ALT and AST, alanine aminotransaminase and aspartate transaminase. They are a couple enzyme. In most requests, they are actually essential for the assessing the liver function. ALT and AST are transaminases enzymatic activity belong to class of the transferases. But about the ALT is more liver specific than AST. ALT elevation persists longer than the AST, formerly named as glutamine pyruvate transferase or GPT. In actually past years, it was actually common writing GPT instead of ALT. And properties about the AST: blood level of AST is increased with many diseases of various organs, like liver disease, myocardial infarction or MI, progressive skeletal muscular dystrophy, crush injury, hemolytic disease. And in artifact, actually, when we are talking that is an artifact increase, it means it comes from the hemolysis sample or serum separation delayed, because our red blood cells are full of the AST, and in hemolysis condition they will release their AST contents to blood circulation. About the formerly name for the AST, we had a glutamate oxalate transferase name, or GOT, like the ALT or GPT for ALT. It was actually historical name for the AST. Nowadays that's the out of date actually term for the AST. We have several causes of elevated levels of blood AST and ALT. But about the differences between how much of this condition cause more increases in AST and ALT in blood circulation, we have highly increase in ALT AST up to 20 to 50 folds in viral hepatitis. In this condition, ALT is much elevated than AST. So viral and toxic or alcoholic hepatitis, cause a highly increase in ALT and AST. What about the cirrhosis? Cirrhosis is a fibrosis of the liver. Actually, it's a kind of end stage of disease for the liver, and that's called cirrhosis. In cirrhosis, we have a moderate increase in AST and ALT in blood circulation, up to 4 to 5 folds. In graph you can see how much of this increase is because of the cirrhosis and or actually chronic disease of the liver. What about the obstructive jaundice? We have a moderate increase of ALT and AST. Actually, the level of increases in ALT and AST is up to 3 folds. And the final actually cause for increasing of this ALT and AST is alcoholic or drug intake in acute condition. We have a transient slight to moderate increase of this coupled enzyme increasing blood circulation. After the increasing level, the other